Hello everyone. In this episode of Learning Pharmacology, I shall discuss presynaptic vesicular function in health and disease. It's form of a PGCMA. Therefore, the target students are post-graduation pharmacology and allied sciences. The nerve terminals, as we all know, of the neurons have a mechanism for synthesis, transport, release, and reutilize neurotransmitters by vesicles. So vesicles are specialized organelles having such mechanisms because of transporters and proteins. The vesicles thus play an important role in neurotransmission. This will be discussed in this. Synaptic vesicles are actually grouped into three pools. The readily available pool is called as readily releasable pool. The second is recycling pool that replaces readily releasable pool, and the third is reserve pool that is activated when there is a high intensity action potential to provide more neurotransmitter reserve. Synaptic vesicles also have two types of carrier proteins. The one is transport protein for transporting neurotransmitters inside, particularly vesicular monoamine transport subtypes 1 and 2. And the second is very important, which is a part of the discussion, is trafficking proteins for exocytosis, endocytosis, docking, recycling, and we will be terms we should know. Trafficking proteins are docking proteins, endocytotic proteins, fusion proteins, priming proteins, and proteins involved in exocytosis. For example, endocytosis is mediated by involvement of several proteins, synaptostagmin and synaptogenin. Synaptostagmin binds with AP2 protein to carry out endocytosis, and synaptogenin is an important protein having phosphatase activity, and that coexists with the vesicular membrane protein endophyllin, and the main function is to carry out vesicular endocytosis for restarting next cycle. Fusion proteins are there, particularly syntaxin and SNAP25, that interact with another protein which is VAMP. One of the VAMP is synaptobrevin. Priming proteins are particularly important are MUNC13 and MUNC18, and proteins are also involved in exocytosis, and they are called a snare group of the proteins that contains SNAP25, syntaxin 1 and MUNCH or MUNCH. These are the terms, these are the acronyms which will be used very frequently, so they have been detailed here for better understanding of the role of these proteins. Now, vesicles exist and function in a dynamic state, and there are 6-step vesicular cycle of formation to recycling, and these are formation of synaptic vesicles from endosomes, docking of synaptic vesicles in the ectozone of the presynaptic membrane, priming of the synaptic vesicles to make them release ready, and fusion of synaptic vesicles with the presynaptic membrane to make the pores, so that there is fifth step is release of neurotransmitter by exocytosis and reutilization of the vesicles for newer cycle is called as endocytosis. Each step will be discussed. For example, step 1 is formation of vesicles. Vesicles are formed from the early endosomes by budding and involves AP2 proteins and clathrin. There are three methods of formation. One is recycled vesicles fuse with endosomes with the help of the proton pump, which that establishes a gradient for uptake of the neurotransmitter and ATPase. Second is new vesicles are formed by budding of the presynaptic membranes, and that is clathrin mediated. And the third is bulk endocytosis. By far, First and second are important. Once they are formed, the vesicles undergo uptake of neurotransmitters by vesicular monoamine transmitter. For example, there will be uptake of acetylcholine or noradrenaline into these vesicles that are stored in the dense granules. In this process, vesicles also require the exocytotic proteins, particularly snare complex of proteins and synaptotecmin. And this will, because these proteins are needed for docking also. Now, docking involves the activation of calmodulin kinase that will phosphorylase synaptin. Normally, synaptin is co-bound with cytoskeletal F-actin, and this must be removed. This must be dissociated to produce the desired docking movement, and upon arrival of the action potential, this removal is done. There is a dissociation that makes vesicle unbound from the cytoskeleton. There is activation and movement of these vesicles laden with neurotransmitter towards the presynaptic terminal membrane is called as docking, so that they become fusion ready. Docking actually results in placement of vesicles very near to presynaptic membrane and depolarization at the nerve ending and entry of calcium initiates docking. That will result in. Next step of the cycle is priming. That undergoes in two phases. One is syntaxin assembles with SNAP25, allowing for vesicle snare protein synaptobrevin to bind to the complex. So such a trimeric complex can be formed, and priming involves another protein called as complexin that binds to this trimeric complex to make it just fusogenic, imagine fusion ready. But still there is a clamp by the complexin. This clamp will be removed for release. You can see here how these steps are produced. Complex formation, role of complexin in priming 2. The next step is vesicle cycle is fusion. Now once fusion occurs, the membranes of the vesicles fuse with the terminal membrane presynaptic, and this involves synaptotecmin that will bind with calcium, triggers the fusion. Still pore is not formed. During exocytosis, now pore will be formed by snare complex of these SNAP25, syntaxin 1 and MUNS18, and is an important role for vesicle exocytosis. Vesicular membrane protein synaptotecmin is another one that acts as a calcium sensor in the event of process of fusion of vesicular membrane with presynaptic membrane. So we can say it is a calcium dependent, and therefore calcium triggers fusion and pore formation. There is a release of neurotransmitter. The next step is with endocytosis. It occurs by two methods. Kiss and run method is faster. Empty vesicles return to new pool, whereas clathrin mediated endocytosis, that is the release of neurotransmitter, the empty vesicles collapse and may involve clathrin, which is a structural protein that facilitates endocytosis. In this process, clathrin binds with the AP2 protein and coats the vesicular membrane, still attached to the presynaptic membrane. Dissociation of clathrin-coated vesicle, as is shown in figure 2, will result in formation of free vesicles. Now, typical example is acetylcholine vesicular cycle. For cholinergic transmission, all elements are shown in the figure. How movement step by step movement results in release of acetylcholine involving trafficking proteins. Botulinum neurotoxins serotypes A and E cleave SNAP25. Botulinum neurotoxin serotypes BDFG cleave VAMP, whereas serotype C cleaves SNAP25 and syntaxin. And because there is a cleavage of these important proteins involved in release, the disturbed trimeric complex assembly will result in lack of release of the acetylcholine, and thus there is a muscle paralysis, longer lasting paralysis. Almost similarly, there is a storage and release of noradrenaline from adrenergic neurons involving the transport and trafficking proteins. Now, let us discuss a little bit about clinical significance of studying this. Vesicular dysfunction in disease. Bacterial infection is one example. Bacteria chlamydia may have snare-like effector proteins that cause accumulation of host proteins in bacteria, and this enables them to drive nutrition from host cells for survival. So they are just camouflaging by utilizing same system which is present in the host. Second is role in neurodegeneration. We will see in details how neuropsychiatric and neurodevelopment disorders, neurodegenerative disorders have some changes in the form of transport proteins and the traffic. In schizophrenia, complexin 1 and 2 are biomarkers for GABAergic and glutamatergic presynaptic neurons in the brain, and there is a reduction in both of these messenger RNA activities, and also complexin 2 protein levels are reduced in the temporal lobe cells of the patients with schizophrenia. Depressive illness, there is a reduced complexin level reported in the neurons of prefrontal and cingulate gyri. Bipolar disorder, reduced levels of complexin 1 and 2 are reported in hippocampus. Alzheimer's disease has so many protein disturbances, in fact called as proteinopathy, but complexin levels are also reduced, and therefore the concept is still evolving, but complexin may also play an important role. In Parkinson's neurodegenerative disease, apart from changes in the neurotransmitters and neuronal proteins, complexin 1 protein is also found to be increased in substantia nigra. Huntington chorea, complexin 2 protein levels are found to be reduced in the striatum, hippocampus, and frontal cortex. Role of syntaxin 1 in epilepsy. Syntaxin 1b regulates GABAergic activity, and mutations are associated with the several types of familial epileptic disorders. Variations in the syntaxin 1b protein contributes to fever-associated epilepsy, and snare motif of syntaxin 1b variations are related to the determination of anti-epileptic activity of the several drugs used. One special protein, synaptic glycoprotein 2A, is a member of protein that is specifically expressed in vesicles and modulates action potential dependent neurotransmitter release in the brain, and is involved in epileptogenesis also. It is a binding site for the levetiracetam and bivaracetam. Therefore, synaptic proteins may also act as the receptors for anti-epileptic activity of some drugs. Missense mutations in SV2A gene is known to cause intractable epilepsy and attention deficit hyperactivity disorder, although occurs in children, but may also occur in adults. And VAM2 protein is also called as synaptobrevin 2 and syntaxin 1A protein, along with the changes in the SNAP25, and these changes due to mutations in VAM2 and syntax 1A genes are reported to increase the susceptibility to such disorder in adults. Variation in the gene causes neurodevelopmental features early in the course of disease, and they can be identified as axial hypotonia, intellectual disability, and features of autism. Vesicular dysfunction in Eaton lumbar syndrome, reverse myasthenia gravis syndrome. Development of autoantibodies against PQ type of voltage-gated calcium channels on the presynaptic nerve terminals is the main mechanism resulting in reduced acetylcholine transmitter release in reverse myasthenia gravis. In addition, synaptotagmin may act as an autoantigen to raise autoantibodies in this disorder. Now, this is in sharp contrast to development of autoantibodies against postsynaptic acetylcholine receptors in myasthenia gravis. So, it is different and is called as reverse myasthenia because of clinical characteristic manifestations. Another is congenital myasthenia syndrome, a rare disorder where presynaptic dysfunction of acetylcholine is shown. It is attributed to genetic variation in the expression of choline acyltransferase enzyme. So, key enzyme CAT enzyme, acetylcholine ACAT enzyme, and that results in reduced acetylcholine release, resulting in myasthenia-like syndrome since birth. Therefore, if try condense the available evidence, the sciences are still evolving and much more will be known in the future. Those who are interested in knowing more can access the information from the given literature. And thank you for the session. You can access more on my YouTube channel. Thank you. Good day.